So I'll go ahead and get started. My name is Ashley. As I mentioned before, I'm part of the prostate work stream. I'm also working on this with Michael, who couldn't be here today. I will admit that he is probably more of the expert on the biology of all this than I am, but I'm going to try to see if I can make things a little bit more clear today regarding the biology, mechanism of action, and some common treatments for prostate cancer. So first, we're going to take a look at the normal prostate gland. So the prostate is a small walnut-sized gland that's part of the male reproductive system. It's located below the bladder and in front of the rectum, and it's surrounding the upper part of the urethra. So its main role is to produce and secrete prostatic fluid, which contains enzymes, zinc, and citric acid. And all of these things kind of help to nourish and transport the sperm. So structurally, the prostate is made up of glandular and stromal tissue. So the glandular part is responsible for secretion, while the stromal part contains smooth muscle that helps to propel the prostatic fluid into the urethra. Um, and so importantly, the growth and the function of the prostate are regulated by androgens. So that's like testosterone, and it has a derivative dihydrotestosterone or DHT. These hormones bind to androgen receptors in the prostate and they stimulate cellular growth and activity. We'll discuss a lot of that later. But in a healthy adult, the prostate remains pretty stable in terms of size and function, but changes to hormonal regulation or cellular signaling can lead to abnormal growth, which can kind of set the stage for things like prostatic hyperplasia or prostate cancer. So this was just a diagram showing the different zones of the prostate gland. This one, I don't know if you can see my cursor, but the peripheral zone is where the vast majority of prostate tumors occur. So that is a lot of focus in terms of research for prostate cancers and potential treatments. So there are quite a few different layers and different cell types that are involved in the prostate. So it's organized into glandular units, which are lined by layers of epithelial cells surrounded by a stromal compartment. So you'll see on here these kind of smaller cells that are kind of lining the outside of this first diagram here on the left, the basal cells. These are the bottom layer of the epithelium, and they sit kind of directly on the membrane, the basement membrane. They serve kind of as like stem-like cells, so they're capable of regenerating other epithelial cell types. And they'll express markers like cytokeratin 5 and P63. The luminal cells, these are the larger pink ones, kind of lining the second layer. Second layer, if you can see here. These are the most abundant epithelial cells. They line kind of the inner surface of the gland. And their role is secretory in nature. They produce the prostatic fluid, and they rely on the androgen signaling for their function and survival. So these are just kind of some diagrams here showing the different cells. We also have, you can see on this right diagram over here, the neuroendocrine cells. These are scattered cells that don't express androgen receptors. Instead, they are in charge of secreting neuropeptides and hormones that influence the surrounding cells and can play a role in prostate development and progression of the disease. And then outside of this epithelial layer is the stromal compartment, which I kind of mentioned before. That's the smooth muscle cells, the fibroblasts, immune cells, blood vessels, and some other components. Its main role is to provide some structural support and communicates with the epithelial cells through paracrine signaling. So that is critical in both the normal function of the prostate and as well as the cancer development. So something else that's
Eric?
important to note is that oh, sorry, was someone going to say something?
Yeah, the term paracrine is something that most people will not be familiar with, I think. Could you spend a second on it?
Sure. So the paracrine signaling, that is a form of communication between cells. It's where a cell releases signaling molecules that affect other cells that are nearby. I think there are four kinds of cell signaling that I at least know of. There's um, depending on the distance of the communication that you're trying to achieve, and the paracrine ones are um, it's meant to be for to target to reach target cells that are within a relatively short distance. Versus endocrine signaling is different. That would be trying to reach targets through the bloodstream that are a little bit further away. So the neuroendocrine is meant to communicate with cells that are relatively close by.
Thank
Okay.
you.
Yep. So I have the two bullet points here. Each region of the prostate contains ducts, and I don't actually know how to pronounce this word, acini, I don't know, where
Asinine.
secretions, asinine, yeah, okay, where secretions are produced by a cluster of cells at the end of an exocrine gland. So that kind of helps with that cell-to-cell communication. So this slide is pulled from the lung cancer presentation that was done a while ago. Just kind of basically goes over tumors and cancers. Um, Figured I would kind of briefly go through this as we have some newer people here, but um, so tumors and cancers are abnormal growths of cells, and tumors can be either benign, which is non-cancerous, or malignant, which would be cancerous. They are clonal and arise from single cells that begin to proliferate abnormally, and so cancers are diseases of these abnormal cells that divide uncontrollably and can invade other tissues. So when cancers spread to other parts of the body, that's when they metastasize. And there are more than 100 distinct types of cancer. So there, just a little bit of information about that. And then this is another slide that I kind of pulled from the lung cancer presentation, just talking more about cancer itself. As we can see, the common word that you're going to hear me say today is mutations. There's quite a few mutations that are involved with prostate cancer, in addition to several other cancers. And the main thing that's kind of happening here is proliferation of the cells when in an uncontrolled way, which causes the tumors and then leads to the progression of the cancer. So we'll kind of get some more into that. Some demographics regarding prostate cancer. It's the second most common cancer in men after lung cancer, which I thought was very interesting. In 2020, there was 1.4 million new cancer cases diagnosed. And in 2018, there was 1.2 million newly diagnosed cases and 350,000 global cancer-related deaths. North America does have the highest incidence rate, but it has the second lowest mortality rate. There were other continents like Africa that, have higher mortality rates due to differences in treatment and availability and access to healthcare and treatments. And the risk of malignancy does increase with age. So in men under 50, the incidence rate was less than 1 per 100,000. Between age 50 and 59, it was 98.3. And then for years 60 and older, it was 508.8. And the main affected populations in the US are men of African or Caribbean descent. They have a 2 times higher risk than white populations, and men of Asian descent in America have a similar risk to white men in America. So then going into the types of prostate cancer, the adenocarcinoma is cancer that arises from the epithelial cells of glands, and most prostate cancers are adenocarcinomas. There are a couple other tumors that are less common but contribute to prostate cancer, neuroendocrine tumors. They're listed there on the screen: squamous cell carcinoma, some other carcinomas, and then sarcomas. There is a difference between the aggression of the cancer. So indolent prostate cancer is slow growing. It grows over a span of several years, doesn't necessarily require immediate treatment. It's not quite as aggressive. We'll get into some of the treatments in a minute here, but the main treatment for indolent prostate cancer would be active monitoring, monitoring the growth of the cancer. And you'll see a lot of adenocarcinomas are this indolent prostate cancer. And then the more aggressive ones, they grow very quickly and often require immediate treatment. to avoid the cancer from spreading to other tissues and other organs. And this will often be like the neuroendocrine tumors, the small cell carcinomas, a lot of times are the more aggressive prostate tumors. So getting into some of the stages of prostate cancer, the staging is similar to how one would stage other types of cancer. So the stage one is where the cancer is small, it's confined within the prostate. Um, it's often found either during biopsy or surgery for other benign conditions. At this stage, the tumor isn't really causing symptoms, and it's not generally a cause for immediate concern. At stage two here, the tumor remains confined to the prostate, but it may be larger. Um, it can be felt, may be able to be felt, and it may involve more than one part of the gland. So it's still localized, but it can indicate that there are more aggressive cells at play. The stage three cancer is where it's begun to spread outside of the prostate into nearby tissues, such as the seminal vesicles, but it hasn't quite reached the lymph nodes or any distant sites. And then stage four is very advanced. It's where the tumor has spread to nearby lymph nodes or other distant organs like the bones or the lungs. And it's at this point that the disease is considered metastatic. Just a little bit more information about the progression of prostate cancer, but taking a look at the cells. So looking here, we see the on the left-hand side, we have the normal epithelium, how I showed in an earlier slide. We get to the prostatic intraepithelial neoplasia. So this is where the cells are now showing abnormal architecture and increased proliferation or that uncontrollable growth. But it still remains confined within the structures of the gland. And as we keep going down the line here, we see that the growth of the cells becomes more and more uncontrolled and starts spilling out until you ultimately get metastasis as the cells continue to grow. And the cancer cells can manipulate their environment by communicating with the surrounding stromal cells. And so they can promote angiogenesis, which is the growth of new blood cells, and that helps supply nutrients to fuel the cancer cells to continue to grow and ultimately metastasize. So the prostate cancer survival rates by age, as you can see here, the survival rate is relatively high, especially compared to other cancers. So I've heard, talking with physicians before, they say prostate cancer sometimes is not necessarily, you don't die of prostate cancer, you die with prostate cancer. It's something that can be very longitudinal. You can see here in men under 60, the 5-year survival rate is 95%. For men age 60 to 70, it's 100. And then for men over 70, it's 85, the 5-year survival percentage. And then, as we get further and further, the survival percentage does decrease, but I would say it's not necessarily, isn't always quite as aggressive, or the prognosis is not quite as, I guess, upsetting than with some other cancers that are a bit more aggressive. So here are some common sites of metastatic spread. We see that there are some common sites here on this left-hand side. The pelvic lymph nodes a lot of times are where we'll get metastasis of prostate cancer. Another one important area is the bone. And then the pelvis, the hip, the axial skeleton. Those are kind of like the more common sites that we'll see metastatic spread and also have a better chance of survival. But these uncommon sites in this text box above, if it spreads to distant lymph nodes or the liver or the lungs or even the brain, while this is more uncommon, it does indicate much poorer chance of survival. Okay, so there's a couple of tools that we can use to diagnose prostate cancer. So the main, I guess the first line of diagnostic tools that are used is the digital rectal exam. So that is where um, you will go see a doctor. There will be palpation of the prostate to assess the gland enlargement, the texture, and its stiffness. If there is an abnormal result, that can indicate the need for a biopsy to test some of the cells that are there. Um, Another test that's done frequently is an analysis of PSA levels. That's PSA stands for prostate specific antigen. This is a blood test that can be a better independent predictor than the exam. And an elevated number would be considered between 3 to 10 nanograms per milliliter. And so the serum or the blood test, the blood serum PSA level is a continuous parameter. It can be elevated, not just to prostate cancer, but also due to things like infection. So a lot of times, the PSA level will be considered relative to a patient's baseline level, and then confirmed with repeated assessment. So you'll see that there's follow-up PSA measurements may be indicated, depending if the patient is at risk, or if you're not at risk, it may be after up to 8 years or so. So the biopsies that we use to diagnose prostate cancer, a lot of times transrectal ultrasound guided biopsies are used for a sampling of 10 to 12 cores for using histology to diagnose the condition. And these samples are typically taken from the peripheral zone, like I mentioned before, the one that has 80% of prostate tumors that lead to cancer are there. So because they're testing mostly from the peripheral zone, these ultrasound guided biopsies can sometimes miss the anteriorly located tumors, but there are also MRI guided biopsies that can help to give us a clearer picture of what's going on as well. So the Gleason system is a way to grade prostate cancer. It indicates how aggressive the cancer is likely to be based on how abnormal the cancer cells appear to be under a microscope. So these scores range from 2 to 10, with the higher score indicating a greater likelihood of the cancer growing and spreading aggressively. So a Gleason score of 6 or less is generally considered low grade. A score of 7 is intermediate, and then 8 to 10 is considered high grade. So if we look on the left over here, this is just a diagram showing what the patterns may look like of the cells. So the first row up here, we have some small uniform glands. It doesn't seem like there's any out of control growth happening. And then with each of the progressive levels going down, we see what looks like more chaos, irregular masses, black or occasional glands down in row 5 here. And then those indicate, so these patterns on the left side kind of show up on the right side over here as we get into the Gleason score. So you'll notice here that in this second row and then in this third row, it'll say 3 plus 4 equals 7, and then 4 plus 3 equals 7. So just because they're the same number doesn't necessarily mean that you would classify the prostate cancer the same way. The predominant pattern is what's stated first. So if we're looking at the second row here, the 3 is listed first. So the predominant pattern that we're seeing is this third row over here on the left side, with the fourth row also prevalent, but not the predominant one. So that would indicate a score of 7, but it's still considered a low grade prostate cancer, versus if the predominant pattern is a 4 instead of a 3, that's where we start to get into the more intermediate risk level. So it's moderately aggressive and can be fast growing. This is just kind of an elaboration of the previous slide. I found some histological images of some of these patterns. I will admit I'm not an expert in reading these, but at least I could kind of, to me, it kind of looked like going from pattern 3 to pattern 5, that there definitely seems to be some more spaces, some more irregularities there. And it I just thought that it would be kind of a good way to represent these patterns and like what they might look like on the cellular level. So there are quite a few treatments for prostate cancer. Um, Going from top to bottom, we have active surveillance. You can do surgery, radiation therapy, um, hormone therapy, chemotherapy, and then immunotherapy. So generally for the localized non-metastatic disease, the main options for treatment include active surveillance, and there can often also be local ablation is the removal or destruction of tissues. So that would be either through surgery or um, your radiation therapy, um, and that may or may not include anti-hormonal treatment, depending. So the treatment decisions would depend on the risk of relapse, which is estimated based on the PSA levels I mentioned before, and then the Gleason score as well. So the general order of operations would be uh, so the surgical removal and the radiotherapy, as I mentioned. And then if the disease continues or if there is a relapse after this initial treatment, the next step would be the use of androgen deprivation therapy or ADT. That's something that I'll mention throughout the presentation. So even though the interesting thing about this therapy is that even though it may be effective at first, almost all patients eventually stop responding after a few years, and that leads to the cancer becoming castration resistant. So from this stage, the cancer progresses and spreads, and it can become metastatic, leading to the need for chemotherapy treatment, as we see here. So a little bit more about the hormone therapy. Um, the main compound at play is called abiraterone acetate, marketed as Zytiga. It's an oral medication used to treat metastatic castration-resistant prostate cancer, and its mechanism of action involves inhibiting a certain enzyme, CYP17, which is crucial for androgen biosynthesis. And by blocking that, it reduces the androgen production um, in the testes and the adrenal glands and in the prostate tumor itself, which limits the ability of the androgens to fuel prostate cancer growth. And I'll kind of mention that a little bit more as we progress. But this androgen production and the androgen pathways are important to the mechanism of action of prostate cancer. So this is just another slide to kind of show what I was talking about, where on the left-hand side, we see surgery is kind of the first treatment that's done as the tumor grows. So as the tumor grows, you have surgery, tumor growth kind of declines, and if it does come back up again, then we move to androgen deprivation therapy, brings us back down to castration resistance. But as I said, this therapy patients eventually become resistant to, leading to metastasis, and then that would lead into chemotherapy, as we have over here on the right. Okay, so talking about some common treatments for prostate cancer, the bullet points on the left are the chemotherapy drugs that are typically used for prostate cancer. But first, I'm going to talk about some of the other treatments that are not chemotherapy drugs, but still important nonetheless, have important targets that are affected by prostate cancer. So the first of which is denosumab, this one here in the third row. So when it's used in prostate cancer treatment, it primarily works by inhibiting the activity of osteoclasts, which are the cells that are responsible for breaking down bone. So this helps to reduce bone resorption, which is the process of bone loss. And in prostate cancer, this drug is useful in managing the bone-related complications associated not just with the disease, but also with androgen deprivation therapy, which can definitely weaken the bones. So a lot of times this treatment will also be given to mitigate the effects of that. So its target is RANKL, Rankel receptor activator of nuclear factor kappa B ligand. And that's a protein that plays a role in the formation and activation of those osteoclasts. It binds to RANK, which is receptor activator of nuclear factor capital B, which is a receptor on the osteoclast, and that stimulates the bone resorption, or the process of breaking of bone loss. So by the denosumab binding to rankle, it blocks its interaction with rank on the osteoclast, and that reduces the rate of the bone breakdown, which reduces then the bone loss that's the side effect of the androgen deprivation therapy. So another drug that I like to talk about is the enzalutamide. That's kind of the 5th row on this table I have here. So this one's important because its target is AR, which is an androgen receptor. I'm going to mention that a few times here throughout the rest of this presentation, but so enzalutamide is an androgen receptor inhibitor. It's used to treat advanced prostate cancer, and it works by blocking the effects of androgens, which, as I mentioned, are the male hormones that play a role in the growth and spread of prostate cancer cells. So it works through competitive inhibition, which is essentially where the enzalutamide mimics the target that so it competes with the androgens to bind to the androgen receptor. It looks similar, has similar properties to the androgens, and so it tries to kind of beat the androgens to the androgen receptor, which effectively prevents the androgens from binding to the androgen receptor, and then prevents that receptor from moving to the nucleus of the cell. So then it inhibits the ability of the receptor to bind to DNA and regulate genes that promote the cell growth in prostate cancer. And that is, as you can see on the right, it says AR antagonism. It prevents the signaling that's part of the AR pathway, which we will discuss in a couple other slides. So talking about some of these chemo drugs, I'll also talk about them in the next slide as well, but the first-line chemo drug that is used is called docetaxel, or Taxotere. It's often given with prednisone. The next drug is cabazitaxel. It's used if docetaxel is no longer effective. And then sometimes older chemotherapy drugs will also be prescribed. These are mitoxantrone and estromycin, I guess. Not quite sure how to pronounce those, but that's what we've got. So the general mechanisms of action for these chemo drugs. So the first two I discussed were mostly concerned with the stability of the microtubules. So the docetaxel will bind with the beta tubulin subunit, which is the protein part of the microtubules, which are involved in the cell cycle. So the docetaxel promotes the assembly of the tubulin into microtubules and then stabilizes them. The microtubules are supposed to be able to depolymerize or kind of break down. So when the docetaxel disrupts that and it stabilizes the microtubules, it prevents the proper formation of the mitotic spindle, or the microtubules that pull the chromosomes apart in cell division. So then that causes mitotic, so it arrests the cell cycle, and it prevents the ability to accurately separate the chromosomes during mitosis. So it arrests the cycle primarily in the mitotic phase, the G2 or phase, which is part of intraphase before mitosis, and then also within mitosis. And that leads to cell death, often through a programmed cell death called apoptosis. And so the cabazitaxel works similarly. It essentially has a similar impact where it stabilizes the microtubules instead of allowing them to disassemble. And then that also prevents essential chromosome separation during cell division. And that causes the cancer cells to kind of stay in the metaphase or anaphase part of the cell cycle. And it also triggers that programmed cell death, the apoptosis. And so those are kind of the main two that are used for prostate cancer specifically. There are some other older chemotherapy drugs, as I mentioned before, that interact with the DNA. The first one, mitoxantrone, it basically inserts itself between DNA base pairs. It causes the DNA to strand to kind of break, and it inhibits DNA synthesis and RNA synthesis. It inhibits topoisomerase 2, which is crucial for the replication and repair of the DNA. And it can suppress the immune system as well by targeting T cells, B cells, macrophages. And yeah, so that was that one. And then the estramustine is an alkylating agent. So it damages, there's a component of it that damages the DNA and other components in cells that rapidly divide, including cancer cells. It does also disrupt the microtubules by instead destabilizing them, versus the first two drugs I talked about focus more on stabilizing the microtubules, but this drug, the estramustine, actually destabilizes them, and it can cause cell cycle arrest. It halts the cell division in the mitosis phase, and it prevents cancer cells from continuing to grow and replicate. So those are the main chemotherapy drugs that are used. These are some of the genes that I've found, or pathways that are important with regards to prostate cancer. There's some other targets that are being studied, but the main one that I'd like to talk about is the androgen receptor. As I mentioned, prostate cancers rely on androgens to grow. And so some prostate cancers can develop resistance to the androgen receptor targeted therapies by altering the activity. So this can happen through mechanisms like amplification of androgen receptors or mutations. But the main goal with therapy would be to decrease androgen production and to block androgens from binding to the androgen receptors. So something that I thought was interesting is that the BRCA genes, which are very involved with breast cancer, actually also have an indication in prostate cancer. So these mutations to the BRCA1 and BRCA2 genes are often inherited, but they work to disrupt DNA repair. And cells with these mutations are more susceptible to treatment with PARP inhibitors, which stands for poly-ADP ribose polymerase. And so the inhibitor is what blocks a protein called PARP. And if the DNA in a cell gets damaged, the poly-ADP ribose polymerase helps to repair it. But cancer cells also have this protein to repair their DNA. So these repairs allow the cancer to thrive and to spread and continue to grow. So treatment with the PARP inhibitor prevents the cancer cells from accessing this protein and then causes the cells to eventually die. And then a third pathway that's important, this is the PI3K, AKT, or mTOR pathway. It's an important signaling pathway that's involved in cell growth, proliferation, and survival. I have a slide about that later on as well. And when it's dysregulated or not working correctly, a lot of times we see that frequently in prostate cancer. So it's the abnormal activation of this pathway that contributes to the cancer progression, metastasis, and even resistance to therapy. And so targeting this pathway with inhibitors has shown promise in both preclinical and clinical studies as a strategy to combat prostate cancer, and especially in cases of the castration-resistant disease, as I mentioned before. So we'll talk a bit more about this. There are a couple key players in here that I will show in a later slide as well. So there are a couple biomarkers we can test for. These are help to guide the targeted therapy options. As I mentioned, it's possible to use genetic testing for the BRCA mutations, the PSA levels, and the blood test I mentioned before is another way that we can test that. We can also test for changes in other genes, such as PCA3 and TMPRSS2 or /ERG. These are urine-based biomarkers. So PCA3 is a non-coding RNA. It's been studied that it's overexpressed in prostate cancer cells and can be detected in urine samples after the digital rectal exam. The TMPRSS2/ERG, that's a fusion between two genes that also tends to occur in prostate cancer cells. And these biomarkers can help to improve the specificity of prostate cancer detection, especially when combined with other clinical information, such as the PSA levels and the BRCA mutations, as I mentioned before. So prostate cancer, as I've kind of been saying, can become resistant to treatment, specifically to the hormone therapy, which causes the castration-resistant prostate cancer. And there's a couple reasons why this can happen. The mechanisms of resistance, as has been studied, can happen through changes in the androgen receptor. So the androgen receptor, or AR, remains active in the castration-resistant prostate cancer cells, even when there are therapies that are designed to inhibit it. The cancer cells can actually develop changes in the androgen receptors that allow it to evade or avoid the effects of these treatments. There can also be changes in the signaling pathways. So prostate cancer cells can alter the pathways, the signaling pathways that involve the AR, allowing them to keep growing, even if there are even in like the absence of androgens. And there can also be an upregulation of steroidogenic enzymes. These are enzymes that synthesize androgens. They can be upregulated or increased, which leads to increased production of androgens within the tumor itself, even when the androgen levels in the body are low. So there's a couple workarounds here, despite the treatments that we try to use. So now we're getting into synthetic lethality. This is a slide that we also took from the lung cancer presentation. This concept is still kind of new to me. I will say Michael is probably a bit more of the expert in this, but synthetic lethality essentially refers to a genetic interaction where the combination of two mutations or gene defects leads to the death of the cell, while neither mutation just alone or by itself is necessarily lethal. So in other words, it means that when two genes, like gene A and gene B, are individually functional, the cells can survive. But when either both genes are mutated or disruptive simultaneously, the cells die. And it mostly arises when two genes play essential roles in the same cellular process or pathway, and the loss of both genes disrupts the process, which can cause cell death. And so identifying and targeting these pairs exhibits synthetic lethality, and it's possible to develop drugs that will selectively kill cancer cells that have these gene pairs while protecting some of the normal cells. So that has already been studied and is continuing to be studied in prostate cancer. This is an excerpt of an abstract that Michael contributed to the presentation. So it is definitely something that is being looked into to try to develop more effective treatments that mitigate the effects of attacking the normal cells surrounding the cancer cells. This is another slide that we took quickly from the lung cancer presentation, just about oncogenes and tumor suppressors, both important players in prostate cancer. So an oncogene, as it says on the screen, is a gene that has the potential to cause cancer. In normal cells, it's referred to as a proto-oncogene. These oncogenes promote cell proliferation or cell growth, and they inhibit the programmed cell death or apoptosis, as I mentioned. Some important oncogenes to note in prostate cancer is AR, the androgen receptor gene, as I mentioned before, MYC, ERG, and ETV1. I'll spend some time talking about those, but there are a couple of other oncogenes that are being studied as well, as you'll see there. So in normal prostate cells, the androgen receptor acts as the tumor suppressor. So it does help to prevent that uncontrolled cell growth. In prostate cancer, however, the androgen receptor undergoes a switch. So it becomes an oncogene where it wasn't before. So it promotes tumor development and progression. And from what I saw, the reason why this happens is still being studied. Um, but it does, the androgen receptor continues to play an important role in the castration-resistant prostate cancer, even after the androgen deprivation therapy. And so it's mutations to the androgen receptors, as I mentioned before, and some other mechanisms that allow cancer cells to bypass the effects of the hormone therapy or, develop that resistance, as I mentioned before. So the next one that's important to discuss is MYC. It's an oncogene that is frequently amplified in prostate cancer. Its overexpression is observed in a very significant percentage of tumors. It's a transcription factor, and it regulates the expression of many genes that are involved in cell growth, proliferation, and apoptosis. And then ERG and ETV1 are similar. They both belong to the ETS family of transcription factors. So they're also responsible for regulating cell growth, proliferation, differentiation, apoptosis. And in prostate cancer, they're also found to be overexpressed due to rearrangements in the chromosomes. And then, as I mentioned before, the fusion between the ER gene and the TMPRSS2 gene is typical in prostate cancer. And it's found in roughly 50% of cases, is what I was able to find. So then, in addition to these oncogenes, we also have tumor suppressors. Their role is to negatively regulate cell division or promote programmed cell death. When tumor suppressors are inactivated or non-functional, that can lead to the uncontrolled cell growth and division that we see with cancer. So the main tumor suppressors at play with prostate cancer, there are quite a few, but the main three that I'd like to talk about are TP53, RB1, and PTEN. So TP53 is a very important regulator of cell cycle and apoptosis. Mutations in TP53 are more often associated with aggressive and advanced prostate cancer, and its inactivation can lead to uncontrolled cell division and tumor growth. So the next one, PTEN, I'll talk about. It acts as a negative regulator. It's part of the PI3K AKT signaling pathway that I mentioned before and will mention again. It's involved mostly with cell growth and survival. So the loss of function of this gene is linked to the development and progression of prostate cancer, and it's found in a significant percentage of advanced prostate cancers as well. And then the RB1 gene, that's also one that's important with lung cancer. It's involved in the cell cycle regulation. And inactivation of RB1 is common in prostate cancer, and especially also in the advanced stages, we see the inactivation of RB1 and the uncontrolled cell division and tumor progression. So this is another slide I took from the lung cancer presentation. I thought it was a very good diagram to kind of show the functions of the tumor suppressors and the oncogenes. So we see on the left-hand side, in normal genes, the tumor suppressors are kind of like the brake. They can stop cell growth as needed to prevent tumors from happening. Our brake is functioning well in healthy tissue. The proto-oncogenes, as I mentioned, the oncogenes are what promote cell growth. And so we have this accelerator, but it's mostly off. It's not necessarily something that is stuck in like the on position, as we see on the right-hand side. In these mutated genes, we don't really have a brake. The brake or the tumor suppressors that we would have that limit the cell growth is defective. It's not working correctly, and the oncogenes are kind of going into overdrive. The accelerator is jammed. We have this excessive cell proliferation that's causing cancer. So I realize this diagram is kind of hard to see, but there are quite a few significantly mutated genes in the localized prostate cancer, so the non-metastatic lower grade prostate cancer. On the bottom left-hand side, the main genes that stuck out to me were ERG, PTEN, and MYC. These are very commonly mutated genes in the localized, less advanced prostate cancer. And then if we come to the next slide, these are the significantly mutated genes in the metastatic, more advanced prostate cancer. And on the bottom left-hand side, AR is definitely the most mutated gene and the highest proportion of patients. It's about like 0.7-ish. So AR is very commonly mutated. We do also see MYC here and PTEN and ERG, but we also see the tumor suppressor genes, the TP53, RB1, are also very highly expressed here as well, in addition to BRCA2. All of these are kind of on this left-hand side over here. Okay, so now I'm going to talk a little bit more about the pathways that are significant with regards to prostate cancer. So this is a diagram of the PI3K, AKT, mTOR, and PTEN pathway. So PTEN, that's the tumor suppressor, as I mentioned. It shows, this diagram shows the role that it plays in regulating the conversion of this yellow circle over here, PIP3 or PIP3 to PIP2, which is right here as shown by this red arrow, and preventing the phosphorylation of this AKT over here, which then prevents irregular activation of mTOR, and then dysregulating cellular functions. So if we kind of go based on the letters that we see here, starting at A, one of the functions of PTEN is that it regulates the cell migration through protein substrate dephosphorylation, which leads to cell, these are pointing here to cell survival, proliferation, and migration. So if we're moving over to B over here, PTEN inhibits the PI3K AKT signaling cascade, which leads to the cell. So if this were allowed to continue, it would lead to cell survival, proliferation, and migration of cancer cells. So moving down to C over here, PTEN actually does interact with TP53, and it can result in cell cycle arrest by increasing the stability of the TP53. So if the PTEN is mutated, it is not able to do that and it is more difficult to cause cell cycle arrest. And then same over here with D, PTEN can preserve the stability of the chromosomes through the centromere, which is this center connection here, this darker circle, which helps to hold the chromosome together. So by the PTEN helping to stabilize the centromere and stabilize the chromosome, it can lead to cell cycle arrest. So when PTEN doesn't function as it's supposed to, it leads to alterations in these pathways. In healthy cells, it works well as a tumor suppressor, and it essentially, as I showed up here, it allows for the conversion of the it antagonizes the PI3K enzymes over here, which this PI3K is what converts the PIP2 to PIP3, and that leads to the activation of AKT, which then activates mTOR, and it then allows for cell survival, proliferation, and migration, as we see here. Another important pathway is the AR pathway. This one seems to be less complicated from what we can see here. So as I mentioned, the binding of androgens to androgen receptors is what instigates, in normal cells, this instigates the individual's reproductive development. It's important to have in normal cells. So it maintains several mechanisms for males, such as spermatogenesis, libido, bone mineral density, and muscle mass. So the healthy prostate epithelial and stromal compartments are what express the androgen receptors. And in cancerous prostate cells, it's the regular androgen receptor signaling is disruptive, and then the proliferative AR signaling occurs. It kind of goes into overdrive. So studies show that cancer growth in the prostate depends on testosterone. As we see up here, this TSD stands for testosterone. It gets converted into dihydrotestosterone through a reduction reaction. And the dihydrotestosterone is what binds with the androgen receptor, as we see here. And it leads to the translocation or the movement of this complex into the nucleus from outside of the nucleus into it, and increases the cell cycle genes transcription rates. So that as these cell cycle targets, as there's an increase in their transcription, it can lead to the proliferation of cancer cells. So the process of the prostate cancer cell proliferation depends on testosterone, its conversion to dihydrotestosterone, and how it binds to the androgen receptor, and then moves to the nucleus, causes transcription of the cell cycle targets, and then results in cancer cell proliferation. And the last main pathway I'd like to mention is the ERG pathway, and kind of how it works along with the PTEN pathway to contribute to cancer cell proliferation. So, as I mentioned before, the ERG is a part of a family of transcription factors. They stimulate cell growth to take place. But in prostate cancer, they're found to be overexpressed. And so, as the ERG, as we see right here, has a fusion with the CMPRSS2 gene, which is that this gene is regulated by androgens, it can lead to overexpression of the ERG. And then that leads to when the target genes by these transcription factors are overexpressed, it can lead to what we see over here with proliferation, anti-programmed cell death or anti-apoptosis, instability of the genome, and all of these things that are listed here, which shows the progression from the normal prostate to ultimately prostate cancer. So yes, as we can see, it leads to uncontrolled cell proliferation, increased angiogenesis, which is the formation of new blood cells that nourish the tumors, ultimately to invasion, and then metastasis, which is the spread of the cancer cells. And that's all from me.
Well, thank you, Ashley. That was fantastic. Really just exceptional work. We'll take a second to give you a little applause. So great job. I do have a few questions or comments. So why don't we go through that?
Sure.
First of all, what is a gene fusion? That's just not a term I'd seen much of. Or Paul, if you know that one.
Gene fusions are usually a type of DNA damage that arises from a piece of DNA being spliced out and then reconnected, where you have the two genes being juxtaposed to each other. Normally, you splice out a piece, and the repair process is put it together, and when they put it together, they just run the two genes together because originally they were juxtaposed. It's a very common, well, not very, it's relatively common in cancers for cells to do that. So it's not just specifically having to do with prostate cancers. There's other cancers that will undergo the same process.
Chris in the chat asks, other risk factors aside from age and gender. I'm just pulling up some on chat. Looks like chemical exposure can be one, pesticides, industrial pollutants, pollution. Is anybody else aware of anything else that is a risk factor for prostate?
Through my research, I also see some risk factors like diet and obesity. So diets that are high in red meat and high fat dairy products a lot of times can increase the risk of prostate cancer, as well as obesity is linked to a higher risk of a more aggressive form of prostate cancer.
Thank you. I think I wanted to talk a little bit, like doing a little bit of a retrospective on some of this and how this relates to, a lot of what we're looking at. So I think one of the key ways that I got was definitely like we need to be very clear what kind of prostate cancer are we supporting? Are we researching, trying to propose for? Because it seems like there is a period of high survival, but there's also a period of potential malignancy. And I think this graphic shows kind of the periods. It sounds like and correct me if I got this wrong, but it sounds like a very common early approach is the wait and see approach, just that, the monitoring, the active monitoring approach, active surveillance here. And that would be an early leading into that androgen deprivation therapy, which it sounds like, to be honest, it sounds like a majority. I don't know if we have a sense on that, but it sounds like a lot of people are coming through that front door as far as like a first line therapy, presumably because it's relatively low side effects. It's not a surgery. It's not a severe toxin. I'm guessing that's why it's so common up front. And then you get to a stage with the castration resistant, and you enter a new milestone where it may still be benign, but we're kind of edging towards metastatic. And I think that's the metastatic and the castration resistant are probably where the chemotherapies are starting to play that role. I mean, it's shown here in the graph is like, when those aren't working, right? You try the androgen deprivation and then the tumor's growing anyway, you move on to the chemotherapies. But, it's important for us to realize that for the majority of for the majority of the cancers, this, you might not want to do a chemotherapy, right? And so if we are researching chemotherapies, we should be clear at what stages might we want to apply this. Now, there are reasons why you might want to apply something earlier, and there's also interpretations to that. If we were to apply other therapies other than androgen deprivation early, like if we are talking about the earlier, more wait and see style of therapies, what would you want to prescribe? You'd want to prescribe something less toxic. You don't want to, you're looking at, the doctor is doing a risk comparison analysis, and they don't want to give you something that's doing more harm than good unless they have to. If it's metastatic and your life is threatened, of course they're going to give you these harsh toxins. But prior to that, they're going to try and not do that. In our research, we have sometimes found that, especially looking at drug repurposing data, sometimes we can find treatments that are already approved and perhaps low side effect profile. And those might be a good fit or a good alternative to androgen deprivation therapy, or maybe something like immediately after that, try this other thing that's like not so bad. But then you enter like a different time period when it becomes life-threatening, and your risk profile changes, where now let's bring in the more severe toxins. Let me pause there if anybody wants to comment on that. Paul, do you feel like I captured that okay?
I had a comment.
Yeah, go ahead, Jackson.
So we did talk about the indolent cancers, which were the slow-growing ones that are observed. But I thought we also mentioned that other forms could be aggressive. So I'm kind of guessing that the wait and see approach isn't 100% of the time. And I get with cancers, it's all very complicated. But it does seem to me that another option would be able to focus on those more aggressive cancers. right?
Yeah, at least being clear what we're targeting and why.
Mhm.
Paul, I'm curious how you would approach the framing of the types of prostate therapies and when they might play a role.
All right. I agree with Jackson in that since these prostate cancers tend to arise in older folks, older men, and predominantly are indolent, wait and see is not a bad approach as long as it's not, as the symptoms do not require something like either surgery or radiation therapy to alleviate side effects from the cancer itself. But the tumors, for the most part, are relatively slow growing. So if you have a prostate cancer arise at 70, which is not uncommon, and most guys are probably not going to live until their late 90s. Wait and see is a relatively good approach. On the other hand, if it's aggressive, you've got to try everything that you can quickly, relatively quickly.
Yeah, so it's like
the ultra aggressive therapies, the standard chemotherapies, like the taxanes, which were mentioned. They're very toxic. You're going to have lots of complications. So you want to try to be in the ballgame before that if you can.
go ahead, Chris. What's your question?
Well, this is a little off tangent and more broad, but then one of the questions in the insurance field was similar to when people graduate out of high school or when they're applying for, high school sports, they have to get a physical. But then at what age should people get a thorough checkup? For example, at age 40, or, as Paul was mentioning, that some of these effects don't happen until 70.
Well, most of the time, I'm 76. I've been getting PSA tests just as a part of my routine annual checkup for, I don't know, at least 20 years. And the doctors frequently don't even tell you, they just run it. And they don't tell you that the outcome if the ranges are within normal ranges.
So Paul's referring to a blood test, Chris. And yeah, he's right. Those probably will just come into your standard of care. I think the prostate exams, I don't know if they still do them. I think they still do at around 50. But then I think they pushed that back a bit. The blood tests have been getting pretty good.
Well, they're good if, again, as Ashley was mentioning, if they're used in perspective, so that you look at linear results over time.
Yeah, that's right.
So if you see a continued increase over time, then those become more concerning. But a single event could be due to, as Ashley was mentioning, a number of factors, including just an irregular infection. So there's a lot of false positives associated and false negatives associated with the PSA test. And following trends again is really the best way to sort of look at the results. And the same thing is true with the therapy. Once you do a therapy, following the PSA trends are going to give you pretty good indication of whether the therapy itself is working, but not just a single isolated evaluation, PSA evaluation. You want to look at trends over time.
Yeah, that's right. And Matt, what you were talking about with the prostate exams, those are kind of like, you're right that they happen like at, around 50, if you're like of average risk. If you like have family history of prostate cancer, then those exams happen a bit earlier in your life, probably like in your 40s. But they are kind of meant to be like, I mean, the PSA tests, looking at the trends of that is a bit of a stronger indicator. The prostate exams are kind of like they help to raise the flag if it's something that, we need to keep looking into, but it's the PSA levels over time that are overall, those trends are a better indicator of the risk of developing prostate cancer. So they're used in conjunction to try to create a clear image as possible to try to create a good evaluation of a patient's risk.
Thank you. Let's talk about some of the opportunities that we have to study prostate and some of the approaches that we've historically done. What I think we have historically focused on are the roles of tumor suppressors, which you have so cleanly called out here as far as 6 key ones. And I think maybe Paul, you could just give us a summary on why tumor suppressors has historically been something we've looked at. Paul, you're...
Tumor suppressors, part of it is just a little bit fortuitous because we've been working with a data set which monitors gene mutations and tumor suppressors. When they have 2 biallelic mutations in the same gene and their tumor promoters, they are non-functional. So we have a clear off signal. So there's no, and that's true with many, if not most of the tumor suppressors. So that provides us a relatively clear signal for which we can look at evaluating drug responses.
Thank you. Let's take a pause, Jackson, to answer your question. What's your question?
Yeah, I was wondering, for a cell to become cancerous, does an oncogene need to become activated as well as a tumor suppressor deactivated? Or is there different combinations of these genes that need to be active, or does it only take a single mutation for a cell to become cancerous?
It could be as little as 1 mutation of either.
Okay.
For malignant cancers, though, you're just going to tend to see an accumulation of mutations. Mm-hmm. That's where, like, TP53, which is the guardian angel gene, it does DNA repair, very commonly mutated in metastatic cancers. And that's you're generally going to acquire that because there's evolutionary pressures for that. But as far as like the origin, when do you get your first benign tumors? They've demonstrated some down to just single point mutations in specific parts of proteins is enough sometimes.
Yeah, I would like to continue with that to say that that extends now to something that you might hear in at least some scientific circles called the 2-hit hypothesis, which involves a continuous series of mutations with a minimum of 2 leading to an actual oncogenic change. So you go from a benign to a true cancer with a minimum of at least 2 different mutations. But Matthew is correct in saying that it all starts from 1 as part of the progression, and that progression can go from 1 to 2 to quite a number of different mutations as the tumor progresses in its metastases endeavors.
All right, thank you both.
Yeah, and keep in mind that there's subtlety to all genetics. You could have things that make it 60% more likely, 40% less likely, and then you sum up all those likelihoods, and you can have a net effect. So it's not always there's we try and do smoking guns because it's more diagnostic, it's easy to target, and it's an easy story to say. But one of the things I want to get up to coming up is there is a more modern role for large data sets and more comprehensive views at what's going on broadly in genes. Often there are big driver mutations, but that's not to say that the non-drivers aren't having any effect. And I think that's something that we're still trying to figure out. Often the therapy will target one part of the pathway, but also often you might be given more than one combination of compounds that will try and target different parts of the pathway, just so you're having a larger effect. And that matches the biology of more than one pathway may be involved at any one time. So I wanted to focus in on these tumor suppressors because that relates to our prior work. So if Ashley, I at one time I had shown you the tool that we use, which is a Streamlit web application written in Python that shows you the top compounds per cancer type by gene mutation. And it was really historically only used to select genes that were tumor suppressors. And so, for example, we already have we have data that comes out. Well, we have an analysis that comes out that shows the sets of compounds that are most effective when you have the TP53 mutated or when you have RB1 mutated. And currently that is done on a gene by gene basis in the raw data. At this point, that has been extended into our data warehouse. So now we've solved all of prostate as well as all the other cancers that we have data for. But we only have taken it to the point of saying when you have a mutation, this one compound is more effective. And so that's useful as a starting point, but we want to jump off further from that. And what we've done historically is tie it to classes of mechanisms of action. You talked about certainly PARP inhibitors as a class. I know that you talked about let's see. Yeah, PARP inhibitors. We talked about androgen mimics and blockers. And then, probably implied here is like BRCA inhibitors, maybe mTOR inhibitors, all kinds of different mechanism of actions that are similar. And so one of the reasons we did that is because, whereas we have a lot of cell lines, we often don't have them all clearly broken out by the gene that we need mutated, right? So we're trying to get larger sample sizes just by looking at classes of mechanism of action. So one of the things that we might want to do, for example, we know that topoisomerase inhibitors are a class that's commonly used, PAP inhibitors. Maybe we can find all of the compounds that are these key inhibitor classes and just generally see what they're doing. And also, especially if we do it computationally, what are some of the inhibitor classes that aren't discussed, or some of these other ones? And it's actually also copied. It's also related to these two slides, which I thought were really fantastic. They show all the commonly mutated genes. And just for background, when we did our lung cancer paper and we spent so long talking about the RB1, the TP53, the reason we did that was we picked genes that were the most commonly mutated, and we built our narrative around that. And so likewise, because you've identified the top compounds that are mutated, we can build a narrative around that. And one of my questions for the group is, there's also this other genes. I mean, you see all, like, say there's like 40 here. There's 40 other genes already identified that we can just grab and use. What kind of analysis would take advantage of the fact that we have response data of compounds for each of these genes? Right? Like, at least in theory, there's probably a lot of holes in that. But say we have 60% coverage. We have a lot of these that have like the actual drug response data. We have that. It's summarized in our S prime value, the pooled delta S prime value. It's a single value, and it shows how effective a drug is. And if you look at it, the mechanism of action level, then how effective that mechanism of action is on average. And so I want to kind of use the opportunity to guide Ashley as far as we could, or maybe to bring Paul in here, we could probably start following the narrative chain that we used in our lung paper, where I think you started. Well, why don't you, Paul, tell us how you built the narrative in the lung paper? What were some of the early and foundational analyses that we did up front?
God. Well, the lung paper was based on the observation that TP53 and RB1 were the two most common tumor promoters in lung cancer as a whole. That includes all types. And that was sort of the core basis of it. We wanted to see if there was any kind of the reason that those are particularly interesting is because RB1 is situated a little bit upstream of TP53, but it can feed into the biological activity and help control the biological activity of TP53. So they're related. And because they are related, we were hoping to be able to make some connections relative to the types of drugs that would be useful in treating one or both of them. And part of the idea was that since RB1 was upstream of TP53, TP53 would be much more resistant with a mutation to most drugs, and that actually did appear to be borne out. The other thing that we noted, but did not report in much detail, was that PTEN frequently had more opportunities for synthetic lethality than the other two. And we didn't make much of it because it was a single cancer, and we wanted to do some additional work, which we did by the way, in the endometrial cancers, and it appears that may be being borne out as well. If that is the case, that may make it very useful, a useful concept to explore as well in prostate cancers.
Thank you. So I think, just to summarize, Ashley, I think that there is a good fit in the original approach in the lung cancer that we can reevaluate. And maybe the breakout item there is to work with Paul a little bit and to think through what would we want to move into in the prostate work stream, as far as now that we've identified some key genes, we know a little bit about the interaction, we know a little bit about the disease. We can talk about what do we want to target, the benign or the metastatic. But I do want to also we can recreate the lung work to some extent, and then Paul is raising a flag that there is something interesting to explore in the synthetic lethal work, which our work is kind of uniquely, it's not unique, but it's one of the very few studies that's well positioned to identify synthetic lethality. It's a pretty new concept, and B, a lot of data sets just aren't set up that way, and ours is by coincidence. And so we have an opportunity to find potential synthetic lethal targets that maybe other studies wouldn't find. And that could be kind of a unique aspect of the prostate cancer work stream, especially since perhaps there's some early thoughts that might be biologically relevant.
The only limiting factor, I think, is the total number of cell lines that we have available, which are that contain mutants of interest. We are limited overall by the fact that we don't have an enormous number of cell lines to work with. We're limited. So if the mutation frequency in those tumors is low, and for example, BRCA and BRCA1 and BRCA2 in breast cancer, the frequency, as I recall, is around 7%. It's not huge. It's very impactful, but it's not huge. If we're in the same 7% of prostate cancers and we have 40 cell lines to deal with, I mean, we may be just constrained by the total number of cell lines that we have. And that's in part why in the lung paper we also, by the way, chose tumors that had very high numbers of expression of mutants in TP53 and RB1, because there was a whole bunch of them. We had numbers with much higher incidence rates.
Right. I wanted to circle back briefly to talk about our data set. So when you open our data set and you explore it, there are a number of cell lines that are flagged as prostate, and then like we know this drug is better or worse than prostate. But what I want to make clear is that prostate is a rough, oversimplified version. As you see, there's many different cell types. We know that the neuroendocrines, actually, what kind of, that was a bad cancer, right? The neuroendocrine ones?
Yeah, I think it led to small cell carcinoma, like more advanced than some of the other ones.
Yeah, that tended to be a really bad one, the ones that advance very easily, and the other ones were more slow growing, if that was the genesis of it. When we talk about we have prostate data, I want to make it very clear that we should assume it's probably a mix of cell types. So we're kind of like squinting our eyes and saying the prostate samples. It's not going to be great, and I want to call that out early. I think it's enough to move forward with a prostate paper, but we need to be very clear that we don't have, at this time, on a per cell line basis, we do not have the distinction on what the cell line origins were. We do have the genetic profiles. Maybe at some point we circle back and do a deep dive on the cell lines in the DepMap data set to like really clarify that, but that sounds like an enormous undertaking, and I'm not sure we're prepared to do that. I want to go back to this concept of what could we do. I think it's clear to me that we could try and recreate the lung paper, which took a kind of a narrative approach to the central, the most commonly mutated genes and what roles did they play. Sorry, most commonly mutated tumor suppressors and what roles did they play in the lung work. But I want to call on some of our computational folks to provide some other thoughts. Maybe, again, something more on the big data or clustering or Bayesian approach. So, Nathan, I want to call on you a little bit. Do you have any ideas on, like, if this was your work stream, where might you want to go with this?
I'm sorry, honestly, not off the top of my head.
Give it some thought. I would like to
hear
your thoughts on that. Not right away, of course. I want to call on also Brian, if you have any thoughts here on like approaches you might take that might include more of the gene sets than just like what we've historically done, which is like manually selecting the like 3 or 4 of the top mutated ones.
Sure. I guess. Do you know in our data warehouse, in the solve table, do we have information on the stage of the cancers that the cell lines were derived from?
I'd have to. My first answer is no. We do not have that. I would have to look into. Maybe we have that somewhere in like one of the data sets I have not looked at. So no, but what we would know, by the way, is we would know the amount of mutations. So I could give you like a frequency mutated indicator of like how screwed up the cell is. because on a range from like 0 mutations to the whole thing is mutated. Like we have 18,000 genes, and I can tell you how many of them are really screwed up.
Oh, okay. Yeah, so I guess like the severity of the mutations this would be useful for sure. And I'm guessing, yeah, like AR and the RB1. I'm guessing this is just indicating the presence of a mutation or any mutation at all in this proportion of patients. But I guess if it's like a stage 4 cancer, there can be multiple mutations.
You are expecting multiple mutations, yes.
Right.
So we would want some kind of like high dimensional approach where it's like, and this is Nathan, I do want to call on you again. You were doing some work on pathways and pathway analysis and Bayesian relationships to pathways. Is there some sense that there's some algorithms out there that can like look at sets of pathways and find the most common sets that are, if we have drug response data related to mutations related to pathways, like are we able to map from response data to commonly useful targeted pathways?
I think that would be, yeah, I mean, referring back to the study that, you and I talked about, I think that that's the sort of thing that would be in their, that rough wheelhouse. That it's essentially a way of kind of going down from, okay, I have a vector of 18,000 genes to I have a vector, of 58 pathways, and then certain things associated with each one, a vector associated with each one, and then that vector can be used to predict response. So it's kind of like it's a two-step solution of sorts. So yeah, I think that this, that approach would let us handle the the small, it helps us bring in some of the less frequent genes that still have a cumulative, effect, a larger effect.
Well, it wouldn't even necessarily have to be a infrequent gene. For example, you frequently find TP53 associated mutations associated with mutations in the oncogene called KRAS. That combination is very common. And both of those genes can involve the AKT signaling pathway. Again, that's very, very common. So you can, just based on those types of threads, you can tie together a series of strings or gene threads starting from the outside of the cell and going to the nucleus, and just monitor those by frequency of mutation. And you just may want to you could, for example, just focus on the on this graph that's showing here, just those that where you have more than 0.2 proportion of the total patients. Those are the highest frequency as a thought process anyway.
I think to summarize a little bit, I don't think we have to answer it tonight. This is an ongoing work stream, but I challenge our computational team to assist Ashley and Michael in thinking about how we can find correlations in our data set. And what I would say is that we should use these genes, like the plurality seen here, and we should also use the tagging. You see how they're color-coded for really interesting things: genomic gain, genomic loss, conversion. Let's throw that in there. Let's throw those as tags and see what we can do as far as that. Some of the work that Nathan mentioned, it won't make sense yet, but hopefully it will in a few weeks, because Nathan is working on a similar presentation on some advanced methodologies around Bayesian inference on pathways. And so this will give us a link between cell lines and drug outcomes through some kind of inferred understanding on pathways. And that'll be very new domain for us, something we're very excited about. But you know, some of that could transfer back down from his work stream, which I can't remember what cancer we're looking at, but it could be reapplied in prostate. And so that's part of the let's work together as a team to knowledge share kind of mandate. Likewise, clustering algorithms, we've gone through some before. If people see an opportunity here to work with Ashley and Michael in exploring some clustering approaches, I challenge you guys to reach out and see what we can do to prototype some of this. Otherwise, our baseline is just let's recreate the lung paper and go through Delta S methodology. But again, I challenge you guys to see what we can do to come up with more. So at this time, I think we could use some other folks supporting Ashley and in her work stream. I know that we have Michael, but Michael's also has time commitments and such, and we could use some other computational folks. So are there any volunteers who come from the computational background that would be willing to kind of switch to the prostate work stream? Brian, would that be something you would be interested in?
Yeah, I guess, yeah, I mean, I have the, we have that table on the data warehouse, and I can just subset for all the prostate cell lines. And I can load that into Tableau and set up a dashboard, I guess, just to start out.
Excellent. Yeah, that sounds good. And then have you done, Felix, help me out here. What was the one that we came down to? It was UMAP plus what for the clustering, the one that had you didn't have to classify all the outliers?
Well, so the clustering, so UMAP is an example of dimensionality reduction. So for clustering, you can do k-means or DB scan would be my two recommendations.
Yeah,
but you have to, you have to first decide what features you want to model within the clustering algorithm. So that's really the first thing that you have to do, and that's where I'm right now on. That's why I'm looking at the pharma guide to pharma data and potential data that we can use to augment our data. Because once we are able to do that, then we'll have more features that we can actually use for clustering. Clustering, I think you recorded the presentation I did.
That's correct. And it should
back.
be on the onboarding website at the very bottom.
Yeah, so really the challenge is just deciding, okay, what features we are gonna be modeling within the clustering. But once you have that, I mean, it's pretty easy to do the clustering. And then the other part is then interpreting the results of the clustering. And there's a whole bunch of ways you can do clustering. I mean, network, we were actually, it was great meeting you yesterday on the Code to Coffee. One of the things that you were mentioning is networks. So that's another example where you're basically just creating this relationships between different data sets. So that's another thing that could complement the clustering. So you may get different insights from like a network analysis versus a clustering analysis that can be used together as well. So the first question is, how do you model? What features do you use to model? And I think whoever is doing the network analysis will probably have to go through that same question. Okay, what am I gonna be doing to measure the level of relationship between one data set or one record to another? Yep.
Thank you. Brian, I'd like to call your attention to this one right here, unsupervised clustering, where we talk about the DB scan. take a look at this one. I believe this is the one that Felix had done for us. And it talks about some of the concepts behind dbScan, which is the clustering algorithm. And then as discussed, UMAP was the dimensionality reduction. See if you can find, A, watch this. B, see if you can find some reference literature, ideally in cancers, but anything, I guess, that would allow us to, basically start the processing, researching the dbScan technique for using in our prostate data, because I believe that one of the things that we can do, and Felix, this would be in parallel, I think, at least the way I'm seeing it, it would be in parallel to the work stream you have, which is another dbScan approach. But I'm thinking Brian can get started on a reduced dbScan where we have just a small number of the features that we already have in our data set.
Sure. I can start out with watching the video.
Yeah.
I've not heard of dbScan, so it will be new to me.
Be new.
Yeah.
Let
Matt,
me,
another thing.
Go ahead.
I'm gonna be posting also my link for my class. I have a lecture on supervised machine learning, which talks about k-means and dbScan, which are two clustering algorithms. If you're familiar with Python, you'll probably appreciate it as well. That one has a little bit more kind of like theoretical. So if you're familiar with Python, how to open a Jupyter notebook, work with GitHub, you'll be able to follow that part. If not, feel free to send me an email and I can, give you some suggestions.
Okay, great. Sounds good.
Awesome. And so just to be very explicit here, Brian, you and Ashley are going to be collaborators. Felix will appreciate any guidance you have as far as educational resources. Actually, we'll connect. I think we have a meeting, or let's get a meeting next week to circle back up, and we'll work on next steps for your prostate work stream. But I think the first thing will just be a data review of some kind. Maybe we do as Brian suggests, start with Tableau, just do some surveys, get some histograms going. Just start to see what is the nature of our data. We had a list of genes, and we don't know how many of those genes we have decent delta S's for. So that's a very easy analysis we can run. How many of those 40 genes do we actually have opinions about because we don't have a data hole? We should be able to find that analysis pretty quickly in Tableau. So let's work on some of that data review. Does that sound good, Ashley, on your side?
Yeah, works for me.
Okay, great. Okay, well, again, huge kudos. That was a fantastic talk. I got a lot out of it. I will post the recording on the website.